All right, Chem 313, Lecture 15. Today we are going to be continuing with our talks of metabolism, and specifically what we're going to talk about today is the citric acid cycle, or TCA cycle is how I abbreviate it usually when I'm writing, and also the electron transport chain. So we are closing in on our main goal, which is to explain how cells obtain the energy to synthesize ATP. And so remember, our overall goal here is to explain how cells get the energy that they needed to synthesize ATP. So where are we going? All right, you've seen this slide a couple of times already. It's showing a flow chart of what happens in order to obtain the energy that cells need to make ATP. So at the top, we have the breakdown of fuels and the oxidation of carbon. And the point of all of that is to generate NADH and FADH2, which are our reduced electron carriers. And then after that stage is done, what happens next is we reduce oxygen, which is a very energetically favorable reaction, and it liberates enough energy that we can synthesize ATP. So today we are going to talk about the last two stages of this process, the citric acid cycle and the electron transport chain. ATP synthesis itself we will cover in the next lecture. All right, so where we have come from so far. We have taken our two major fuel sources that we're going to talk about in this course. So we've had glucose and fatty acids. And what do? Well, glucose gets broken down into pyruvate, and then from pyruvate it gets converted into acetyl coenzyme A. So pyruvate would be this molecule. Acetyl coenzyme A, since I don't like to draw coenzyme A, I represent it just like that. This is the process of glycolysis, going from glucose to 2-pyruvate. Pyruvate to acetyl coenzyme A is the job of pyruvate dehydrogenase. And then last time we said that fatty acids get broken down directly into acetyl-CoA by the beta oxidation process. Now, in the process of carrying out these conversions of carbons, we also get some useful side products, right? So glucose, when it's broken down by glycolysis, will give us just 2 ATP by itself, which is handy. And we said that one of the nice things about glycolysis is that it gives you ATP, a small amount of ATP, without ever having to have any oxygen present. We also get 2 NADH. Pyruvate dehydrogenase gives us some NADH. And then fatty acid beta oxidation gives us plenty of NADH and FADH2. So you'll recall that for a saturated fatty acid, if you have a fatty acid of N carbons, you get N over 2 minus 1 NADH and FADH2s from beta oxidation. So as we're going to see, all of this NADH and FADH2 is going to be fed into the electron transport chain. And that's where we're going to liberate the energy that we use to make ATP. But we're not there yet. And the reason we're not there yet is we still have all this acetyl coenzyme A to break down. And what we're going to see now is that through the citric acid cycle, oxidation of this acetyl coenzyme A gives us even more NADH and FADH2 to feed into the electron transport chain. So the citric acid cycle is the pathway where most carbon oxidation in aerobic respiration takes place. And that's going to be what we talk about right now. All right, this is the citric acid cycle, also known as the TCA cycle, which is how I usually write it, or the Krebs cycle. TCA stands for tricarboxylic acid, and the reason that it's called the tricarboxylic acid cycle is because of the first intermediate in the cycle, which is citrate. That is a tricarboxylic acid. We've got 1, 2, 3 carboxylic acid groups in it. That's where the name comes from. All right, so for all pathways, you're not responsible for knowing the names and structures of all intermediates, but you are responsible for knowing a few key factors. So where and when does the citric acid cycle occur? So it occurs in the mitochondrial matrix of all cells, and it occurs when ATP levels are low and oxygen is present. So the citric acid cycle cannot occur without abundant oxygen in the cell. Its main inputs and outputs. So because this is a circular pathway, it doesn't really make sense to give a complete balanced chemical equation because you end up regenerating what you started with. But we can simplify the inputs and outputs of the citric acid cycle as follows. For every 1 acetyl coenzyme A that goes into the cycle, what we get out the other end of the cycle is 3 NADH, 1 FADH2, and 1 ATP. And as a byproduct, we get 2 CO2. All right, so notice that acetyl coenzyme A has 1, 2 carbon atoms in it that can be oxidized, and in the process of going through this cycle, we release 1, 2 CO2 molecules. All right, so the citric acid cycle is where most of our CO2 ends up being formed. The purpose of the citric acid cycle, there's actually multiple purposes. The main purpose is to oxidize acetyl-CoA to CO2 in order to generate NADH and FADH2. That's the main purpose of the citric acid cycle. A lesser purpose of the citric acid cycle is to generate intermediates for other pathways. So we're only going to see one of those other pathways, but the most important intermediate for use in other pathways would be oxaloacetate. So that would be our purpose, that would be our point. Regulation we will talk about on the next slide. All right, so the names of all the intermediates in the citric acid cycle are given on the screen in black. The names of the enzymes that catalyze these reactions are shown in red. So there are 3 of these reactions that are especially important that you need to be responsible for. Those are reactions 1, catalyzed by citrate synthase, 3, catalyzed by isocitrate dehydrogenase, and 4, catalyzed by alpha ketoglutarate dehydrogenase. Right? Notice that reactions 1, 3, and 4 are the 3 irreversible steps. And they're irreversible for various reasons, but specifically in the case of citrate synthase, reaction 1, it both is highly favorable, so its delta G is extremely negative, and we talked about last time why reactions with very negative delta Gs are irreversible, and there's a homework problem, or actually a worksheet problem related to that. It's also the rate-limiting step of the citric acid cycle because it's the slowest reaction, okay? By the way, the slowest reaction doesn't necessarily mean that it's going to be extremely favorable or irreversible. But in this case, citrate synthase reaction 1 is very favorable and also happens to be the slowest reaction of the whole cycle. Now, one thing you'll notice about this cycle is that we get 3 NADHs, isocitrate dehydrogenase. Once again, dehydrogenase, that means that we're oxidizing carbon and reducing something else. NADH is released by alpha ketoglutarate dehydrogenase and malate dehydrogenase. And then we get our FADH2 at succinate dehydrogenase. So once again, 3 NADH and 1 FADH2 for every 1 acetyl-CoA. We said one of the other products of this reaction cycle is ATP, and it doesn't appear anywhere on this slide. The closest we get to ATP from this cycle is GTP, that's at succinyl-CoA synthetase, but GTP can be converted into ATP by another enzyme called NDP kinase. That's not really part of the citric acid cycle, but we still say that the citric acid cycle gets us an equivalent of ATP. So once again, 1 acetyl-CoA, 3 NADH, 1 FADH2, and bear in mind, all of this is in addition to any NADH and FADH2 that we already got, because remember, where did our acetyl-CoA come from? It came from either glucose or fatty acids, and the process of breaking down fatty acids into acetyl coenzyme A already gave us a bunch of NADH and FADH2. And so what we get in the citric acid cycle is on top of the NADH and FADH2 we already have. All right, regulation of the citric acid cycle. There's 3 regulated reactions, and I'll give you 3 guesses which ones they are. That's right, they're the 3 irreversible steps. 1, 3, and 4. And this slide diagrams how they're regulated. They're all regulated allosterically. So this slide gives you more detail than I care about you knowing. All I care about you knowing is this. Reaction 1, ATP and NADH inhibit, ADP activates. Reaction 3, ATP and NADH inhibit. In this case, the NADH inhibition is product inhibition. ADP activates. Next reaction, alpha ketoglutarate dehydrogenase. NADH inhibits. AMP activates, but if you said ADP, I would give you full credit on that one. All right, so notice the pattern here. What do we notice? ATP and NADH inhibit the citric acid cycle. Does that make sense? Well, it should make sense, because what is the whole point of the citric acid cycle? We're going to generate all kinds of NADH and FADH2. So in other words, NADH is a product of the citric acid cycle. And if there's a ton of that floating around, the cell is wasting its time making more of it. And likewise, what do we need NADH for? In the next step, we're going to oxidize the NADH and use that energy to make ATP. If there's a ton of ATP lying around, we don't need the citric acid cycle to run. Okay? ADP and AMP activate the citric acid cycle. That actually makes sense, and the reason for that is because if ATP levels are low, then ADP and AMP levels will be high. Why is that? That's because what's the whole reaction that ATP uses to power unfavorable reactions? ATP plus water reacts to give us ADP and phosphate. And then there's another less frequent reaction, but sometimes does come up in metabolism. ATP plus water reacts to give us AMP and pyrophosphate, PPI, which is just two phosphates stuck together. So both of these reactions are very favorable, and they can be used to power reactions that would otherwise be unfavorable. So that means that if ATP levels are low, probably that's because we've been doing these reactions a lot, and therefore ADP and AMP levels will build up. So there's this term that's used in the trade called energy charge. And the energy charge you can think of as the ratio of ATP levels to ADP and AMP. If it's high, that implies that you've got lots of ATP. All right, so the citric acid cycle is regulated by energy charge. It's a shorthand way of saying that ADP and AMP activate it and ATP inhibits it. And that's a general feature of catabolic pathways. So catabolic pathways, of which this is one, they tend to be inhibited by a high energy charge. Anabolic pathways tend to be inhibited at a low energy charge. And I leave it as an exercise to you to figure out why that would be. So to summarize, ATP and NADH inhibit the citric acid cycle. ADP and AMP activate the citric acid cycle. All right, we are done oxidizing carbon. So recall that the whole point of aerobic respiration is that we oxidize fuels to give us carbon dioxide. And we're done with that. We got our carbon dioxide completely formed. And what do we do with the electrons that we strip off of carbon? Well, we have electron carriers that are initially oxidized, and with those electrons, we reduce the electron carriers. So this is our NADH and our FADH2. All right, so now we have plenty of NADH and FADH2. So what do we need to do now? Well, now we need to close the circle by reoxidizing these electron carriers. And when we reoxidize them, we liberate energy. So how do we reoxidize these electron carriers? So you can't oxidize something without something else becoming reduced. Electrons don't just go zipping off into the universe. So we need something to oxidize the electron carriers. What oxidizes the electron carriers is oxygen, because that's our end electron acceptor for this whole process. Water is the reduced form of oxygen. So this half, this lower half of aerobic respiration is called the electron transport chain. So the electron transport chain is a relatively short pathway, although the enzymes involved are very, very complicated. But the electron transport chain's function is to oxidize electron carriers NADH and FADH2 and reduce oxygen. And in the process of doing this, we will harvest energy. Now, notice that ATP synthesis is not part of this process. We have not made our ATP yet, and we're not going to make our ATP yet. We have to store this energy in the meantime. And so the energy that comes from reduction of oxygen is going to be stored in the form of a proton gradient or an electrochemical gradient across the inner mitochondrial membrane. So what I mean by that is that we have this intermembrane space, and the whole function of the electron transport chain is as we liberate energy, we are going to end up in a situation where we have a high concentration of protons in this intermembrane space, and in the matrix, we have a much lower concentration of protons. And you guys should remember from your Nernst equation, whenever you have a difference in concentration across a membrane, you have a difference in free energy. All right, so what are these reactions that take place that allow us to generate this proton gradient? So there are really only 4 reactions of the electron transport chain. They're relatively complicated, and I'll walk you through each one of them at the next slide. But they take place on 4 big complicated enzymes that we call complexes. So what's going to end up happening is this. NADH, this diagram, by the way, is showing the flow of electrons. So what's going to happen is that NADH will hand off electrons through complex 1, our first respiratory enzyme complex, to a molecule called UQ, or coenzyme Q, or if you prefer, just plain Q. Okay, this is another sort of temporary electron carrier. At the same time, FADH2 is going to hand off its electrons through complex 2, again to coenzyme Q. All right, so coenzyme Q is a lipid electron carrier. It has a long hydrocarbon tail, and at one end of the molecule it has a quinone ring, something like that. This would be the reduced form. The textbook shows the structure of coenzyme Q. This hydrocarbon tail is embedded in the inner membrane. Of the mitochondrion. All right, so the entire electron transport chain takes place on the inner membrane of the mitochondrion. So coenzyme Q is going to carry its electrons through complex 3 to cytochrome C. Cytochrome C is a protein electron carrier. And then lastly, cytochrome C hands off its electrons through complex 4 to oxygen, forming water as a byproduct. We said that the point of the electron transport chain is to generate this gradient of protons across the inner membrane. So we actually generate the proton gradient as we go. At each stage of this process, excess energy is used to pump protons into the intermembrane space. All right, and that actually happens at 3 of our complexes: complex 1, complex 3, and complex 4. Complex 2 does not pump protons. All right, so with that set up, here's the electron transport chain. Okay, where and when? It is on the inner mitochondrial membrane. When NADH and FADH2 and oxygen are abundant. The purpose is to oxidize electron carriers, reduce oxygen, and use the energy to pump protons, which we will later use to generate ATP. Inputs and outputs. The inputs are going to be 1 NADH or 1 FADH2, and the outputs would be 1 water. I should add 1/2 O2 as an input. However, that's not really the inputs and outputs that we care about. What we care about are protons being pumped into the intermembrane space. So we can consider that an output. And as for regulation, it may surprise you to learn, this is not really regulated. And I suppose that's because everything up to this point has been regulated much more tightly. So at this point, we're just at the final stretch. All right, so there are 4 reactions of the electron transport chain. Reaction 1. NADH becomes oxidized and the next electron carrier, UQ, ubiquinone or coenzyme Q, is what's going to pick up the electrons. So NADH hands off its electrons to UQ, and that gives us NAD+, and UQH2, which is the reduced form of coenzyme Q. And remember, coenzyme Q is a membrane-bound electron carrier, so it stays in the lipid bilayer. In the process of handing off electrons from NADH to coenzyme Q, we get 4 protons pumped from the matrix, which is the bottom of the page, to the intermembrane space at the top. So we get 4 protons from 2 electrons from NADH. Okay? So what I mean by that is that in the process of oxidizing NADH and reducing coenzyme Q, we generate enough energy that the enzyme can physically translocate 4 protons across the bilayer into the intermembrane space. So in other words, complex 1 is both an enzyme and also a transporter. So that's the reaction of complex 1. All right, complex 2 happens completely independently of complex 1. They don't have to happen first 1 and then 2. So complex 2 is like complex 1, but it does the same thing for FADH2. So FADH2 and UQ, we simply hand off the electrons, giving us FAD and UQH2. However, complex 2 does not pump any protons. That's the only respiratory complex of the electron transport chain that doesn't pump protons. All right, so complex 2 is just an enzyme. Next step, complex 3. What happens at complex 3? So we've generated this reduced electron carrier, UQH2, and now we're going to hand off the electrons once again. So the UQH2 is going to hand off its electrons to cytochrome C. And in the process, we get UQ again, plus 2 cytochrome C reduced. And in the process, we get 4 protons pumped into the intermembrane space. Now you may be wondering, why 2 cytochrome C? The reason for that is because UQ, coenzyme Q, just like NADH and FADH2, is a 2-electron carrier. It picks up a hydride ion. Cytochrome C is a 1-electron carrier. So your textbook talks about cytochrome C in more depth than I care to cover. But in the oxidized version of cytochrome C, it has an iron ion that's in the 3+ oxidation state. And when it's reduced, it becomes iron 2. And iron can really only exist in those 2 oxidation numbers. You can't give iron a second electron and have iron 1 because that's not stable for reasons that we won't get into in this class. So because cytochrome C can only carry 1 electron at a time, it takes 2 of them to take the 2 electrons from UQH2. Regardless, this is a favorable enough reaction that we have energy left over to pump 4 protons into the intermembrane space. All right, last, complex 4. We take our 2 reduced cytochrome C and 1/2 of O2, and we react them to regenerate our oxidized cytochrome C and water. And we need 2 protons to balance out our charge. Water is the ultimate waste product. We're now done. We've reduced oxygen, and this is energetically favorable enough that we can pump 2 protons at complex 4. So you may be wondering, why do we write this as 1/2 of O2? Well, what we're doing through this whole process is we are tracking electron pairs, and we're starting off with NADH and FADH2, which once again carry 2 electrons. We hand them off to ubiquinone, coenzyme Q, which also carries 2 electrons, to 2 cytochrome Cs, which each carry 1 electron, finally down to 1/2 of O2, which is a 2-electron acceptor. So the stoichiometry of this, if you wanted to do it on the per oxygen basis as opposed to an electron pair basis, we would have to double everything. It's just easier to say 1/2 of O2. All right, so now we are done with all of our oxidation and reduction chemistry. All right, the whole point of this now is to generate this proton gradient. And that's what we now have. In the matrix of the mitochondrion, which is at the bottom of the slide, we have a relatively high pH. We have a pH that's roughly 7. And in the intermembrane space at the top, because we've been pumping all these protons into it, we have a lower pH. We have a pH of about 6. And so now it's going to be very energetically favorable to allow our protons to leak back across the lipid bilayer. That would be very favorable, and we can then use that energy to do something that would be otherwise unfavorable, which is to synthesize ATP. And that's exactly what's going to happen next. So our topic for the next lecture is going to be ATP synthesis. All right. In the meantime, check out homework. I believe we're up to 6 or 7 now, and worksheet 7, which are going to give you some sample problems related to all of these. You should be able to recreate the 4 steps of the electron transport chain. It's not terribly complicated. And then in the next lecture, things are going to start getting a bit easier when we get into ATP synthesis. So I'll see you guys next week.